My name is Ashley Adams and I'm a tutor at Chegg Tutors. And today I'm going to be talking about multiple crossovers and what really goes on during that process and to clarify things and make things much more easier to understand. So here at this diagram, I'm going to walk you through step by step before we tackle a problem that's right below me. But let's bring our attention over to this first diagram where we have two loci, A and B. And I drew out little diagrams below because that's how I typically like to visualize what's going on when crossovers occur. And it makes sense to me. You might not need it. The diagram might work better for you, but I like to have it as a reference. And whichever method works better for you, great. It'll help you tackle your problems a lot more easily. But I wrote the genotype out on the left, which is big A, big B, little b. And you can see here, this chromosome here has big A and big B on the same side. And then on this white one here, you see little a and little b are on the same side. And when we have two crossover events occur between two loci, A and B, we see here that they pretty much cancel each other out. So that at the very end of meiosis 2, when you look over here, you see that all the gametes are the same as the parental gametes, and that the progeny will have the same phenotype as the parents because nothing really changed. It would be as what you expected to happen, where you would get either big A and big B would be passed down, or little a and little b would be passed down together, and you wouldn't see much difference. And in this diagram, I'm going to draw it out and show you what would happen, and this is kind of how my brain works, is that if a crossover event happened here and then happened again, then they would cancel each other out, and you end up having what you see on the right, where they're still on the same side. If you were to have just one crossover event occur between loci A and B, then you would have things switched around and you would have big A and little b on the same chromosome, and then you would have little a and big B on the same chromosome. That's what happens in a single crossover event. And then you would start to see different combinations of phenotypes occurring. And now let's bring our attention to the three loci model where things are different, where when you have two crossovers occurring, one between the A loci and the C loci, and another between the C loci and the B loci, you end up with something called double crossover, and you end up with progeny that have different phenotypes from the parents. And so when you see here, when these crossover events occur, I'm going to draw it out here as well. When they happen, you get a switcheroo twice. And looking at this, you'll end up with big A, little c, and big B on the same side. You'll have little a, big C, little b on the other side. And you can see it here what happens after when crossing over occurs, and then the end of meiosis 1, you're left like this. And when they separate out in end of meiosis 2, you have the parental gamete, you'll have that double crossover gametes over here, and then you'll have the other parental gamete here. And so that's what, and if these double crossover gametes are inherited in the progeny, then you're going to get different phenotypes, right? And so that's pretty cool to get to see that's how it works and how we see so many different varieties and how many genes are passed down. And so now let's tackle the problem that's right below me. And for example, if a crossover between the A and C loci over here occurs in 20% of the tetrads, and then between C and B loci in 10% of the tetrads in an individual with the genotype ACB capitalized lowercase little a, little c, little b, which is what I wrote over here and which is what this problem is. What percentage of the gametes are expected to be the double crossover types big A, little c, big B, and little a, big C, little b, which is what we see here, or right over here. Draw a little star next to it. So think of it at these crossover events as independent events occurring. So to put it into perspective, if I wanted to make an analogy, it would be the same thing as asking me, what's the probability of me rolling the die and getting a 1 on the first try and then getting a 2 on the second try? Well, the probability of rolling a 1 on just one die is 1 out of 6, right? Because there's only one side that has 1 on it, right? And then the probability of me rolling a 2 on a die, there's only 1, 2, so it's the same thing, 1/6 chance. But if I want that combination, then they're just multiplying those two independent events. So it would be 1/6 times 1/6, and you get 1 out of 36 chance of me getting a 1 and a 2 rolling that die. And so this is the same thing here in this example where you have 20% chance of crossover between A and C. Then if you have 10% chance of crossover between C and B, if you multiply those two together, you'll get the percentage of the double crossover gametes. So I'm going to write that out over here. So if you do 0.2, or 20%, times 0.1, 10%, you get 0.02. So we have a 2% chance of getting these double crossover gametes. And that's how you would go about solving that type of problem to figure out what's the percentage of progeny that will have that these double crossover gametes will occur in. And one more last thing to wrap things up is that these double crossover events are very significant when you have 3 loci models, 4 loci models, etc. But in a 2 loci model you see here, it didn't really do anything. And if we look at this 2 loci model with A and B, if we have any odd amount of crossovers occurring between these 2 loci, so if we had 1 crossover event, 3 crossover events, 5 crossover events, we'll get what we see right here, right? Which is this what it would look like a single crossover, because the odd amounts will end up having a crossover event occur. But if we have even amounts of crossovers between 2 loci, as you can see here with 2, 4, 6, 8, they cancel out each other, so that you're still just going to end up with the same parental gametes that you would expect to see, like here, big A and big B on the same side, and little a, little b on the same side. And so that pretty much wraps up my talk on multiple crossovers. I hope that explains everything, and good luck to everyone.